So this will be the second discussion on the respiratory system. And the next exam, as you know, on Thursday, I'm going to meet and we're going to have a review session. And I'm going to show you some magical things to know. And while I'm all about recording things, I would really encourage those who are sitting in your stockings right now listening to this at home to be here, okay? Because, you know, if it fails, I won't be able to re-record a lecture relating to a review session. So last week we did have that little glitch and it didn't record, and I had to re-record that. So please be here on Thursday. It'll be helpful, I think. And I may give out a few little secrets to those who are here, and those who are not here won't have that benefit. Number two, the exam is going to cover three systems: lymphatic, respiratory, and digestive. This is the second of the respiratory discussions. We finished up lymphatic a little bit last time, mostly respiratory. We'll finish up with respiratory today, I anticipate. I did record already one lecture on the digestive system. It came at 1 hour, and there'll be a second digestive lecture that I'll get done tomorrow morning, and that will give you 2 digestive lectures, 2 respiratory lectures, and 1 lymphatic lecture. So there's 5 talks, 3 body systems. Thursday will be a review session. Next Tuesday will be the exam. And I also have, or had in my hand 5 minutes ago, your case studies. I must have put them in my office. So the case studies look fine. The only issues with the case studies is if you did not have references, right? You can't tell me about medications and things unless you have references. I was very kind compared to most classes. And if you didn't have references, I only took off 10%. Okay, so it wasn't huge, and if you didn't have references for either, I took off 20%. So the lowest anyone got was a 20 out of 25. If you only had references on one of the 2, it was 22.5 out of 25, and that's really the only thing that anyone got dinged on. So just make sure that on your second set of case studies that you cite at least a few of the facts that you include in your paper, and you include that citation at the end or somewhere in the body of your case study. That's the only thing that was a concern, and I expect just the second ones to go. Everyone should get perfect score. Now you know not to forget any report on the top of it. Don't forget your little case study report on the top, that all of you played well in the sandbox together, and then make sure you have citations. Amanda, are you happy with the mastering assignments? I spread them out. I can't remember what you said. I just remember you made a comment about the mastering assignments. I wish they were blank. And whatever you said, I knew I couldn't make everyone happy. So I just knew that you made a point of comment. I put kind of like one thing to do every day. So there's a homework, there's a quiz, there's something. Don't lose track of mastering. I spread it out so that people were being forced to move along. And lymphatic, that'll be the first thing you'll finish up. And then respiratory, and then digestive was at the very end, since those lectures are online. Okay. So 3 systems, 5 lectures on the exam next week. And honestly, I don't expect this to be a very difficult exam. I really don't. I think the material is pretty straightforward. There's not a lot of numbers. There's not a lot of really difficult concepts. So I suspect you'll do really, really well. Just study and know your stuff, and you'll do really, really well. Right? Okay. What else? Anything else housekeeping-wise? Anything else housekeeping-wise? Final exam? Oh, let me ask you about final exam week. Your exam will be on Thursday of final exam week. I believe that pops in on the 30th. Is that Thursday? Tuesday night, exams officially start on Tuesday night. And I have a question for you. Would you want to have a review session on that Tuesday night during regular class time? Or would you not want to have class? That's really the option. It's either not have class or to meet on that, because that Tuesday at 5 o'clock is when officially finals begin. You wouldn't have a final at that time. But I thought I would give you the option of having a review session. Would you like to have one? Again, so we'll have a review session on that Tuesday during class time, and then on Thursday you'll have your exam. Okay, that lab will be over with that week. There'll be no lab that evening. It'll be the only thing you would come in for. There's no classes on that Tuesday. That Tuesday is reading day. So there'll be no classes anyway. It would just be coming in for this review session. Okay, so we'll plan on that. The final exam will be also over 5 lectures. It'll be over the male and female reproductive systems and the urinary system. So the urogenital systems will be the focus of the last and final non-cumulative exam. Okay, fair enough? let's get moving on to respiratory. First of all, are there any questions? Anything that I've said so far? We finished up with tidal volumes and looking at that spirometry diagram. Next week, you'll be seeing more of that in lab. So when you get to it in lab, you'll say, oh, I've already seen this, I know this. It'll be a good confirmation of what we're doing here. But are there anything at all related to structure or to any of the functions of the respiratory system? I'm just flashing through these things, getting down to what we started. We started at 21.7, talking about the type 1 cells and the type 2 cells that are found down in the alveoli. The first 6 or so sections, modules, were review. We talked about that respiratory membrane. Talk to me about that respiratory membrane. What does that mean? Respiratory membrane. If you hear that, it's code for what? Really a triple layer. What are those 3 things that make up, quote, the respiratory membrane? We're talking about gas exchange here. And air has just been brought down into your lungs, down into your alveoli. And those little oxygen molecules need to hop out of the alveoli, hop through the alveolar wall, the simple squamous epithelium, go through a thin layer of basement membrane, and then jump into the capillary, which is also surrounded by simple squamous epithelium, and hitch a ride on the hemoglobin in the red blood cell. So the respiratory membrane refers to that triple membrane epithelium of the alveolus, basement membrane, and endothelium, also simple squamous, of the capillary. So those are the 3 layers, if you will, of the respiratory membrane. Then we talked about external versus internal respiration. If you talk to a respiratory therapist, they're talking about external respiration most of the time. They're talking about how are the lungs functioning, how are they bringing air in and getting rid of CO2. To a biologist, though, or to a medical, in addition to external respiration, the whole purpose of external respiration is to allow your cells to get oxygen. So at the cellular level, at the tissue level, what you're focusing on is the ability of that oxygen to get to the tissues in a timely manner, to provide a metabolism, and eventually that oxygen, through the processes of electron transport, are going to create the ATP. So without that O2, right? Without the O2, you're not going to be able to make the ATP necessary to run your metabolism. So that would be really more that internal respiration. So you got these 2 very much connected portions, and it just depends on who you're talking to, if they're talking about external or internal respiration processes. Hypoxia would be a lack of oxygen going to your tissues. And then we mentioned how, according to Boyle, right? Who was Boyle? What did Boyle say? Where was Boyle's name? Did I pass him? There's Boyle, right? Boyle's law, a physics or a chemical gas law. Boyle said that pressure and volume are inversely related. So as pressure goes up, volume will be going down. Or as volume goes up, pressure goes down. And we discussed how this is true of a balloon. If you squeeze a balloon, the volume is being decreased and the pressure inside that balloon is going up. Or in your lungs, as you inhale, what's happening? Talk to me about inhalation. What muscles are first being used in inhalation? Normal, quiet, non-stressful inspiration revolves around 2 muscles. 75% the diaphragm, right? And I'm saying diaphragm so you spell it right. Don't say it that way. And number 2, the other 25% or so is your, which one? Intercostals, which one? Internals or externals? External? You may want to confirm that? Confirmed? Okay. Voted on? Approved? Okay. So the external intercostals. And that's quiet inspiration. And what's happening as you inspire? The diaphragm is going from a dome shape down. The external intercostals are doing what? Expanding. As a result, the overall volume of your thoracic cavity is expanding. As volume goes up, pressure goes down. Now, as that pressure goes down, what's going to take advantage of that lower pressure? The atmosphere, right? The atmosphere is sitting at about 760 millimeters of mercury. That's at sea level. And that pressure, as the pressure decreases in our thoracic cavity, air is now kind of pushed, if you will, into our lungs. At a higher altitude, where there's less atmospheric pressure, not as much air would be pushed into your lungs as in Denver. Okay, a little bit less air. So there would be some more huffing and puffing going on because you're not getting the same oxygen levels at higher altitudes. Now, what happens if you're going to forcibly inhale? You're going to increase more muscles. What are those other muscles that get involved if you're forcibly inhaling? You can read. What were they? We're going down a few more pages, right? So what were those muscles? Let's go down to those pages. Right? Sternocleidomastoid. Scalenes, you know where those are? Better, right? Little test tonight, maybe, this week, scalenes. And pec minor and the serratus anterior. So those muscles are going to help you forcibly inhale more air. Now, let's think about the spirometry value. I introduced this to you. I'm going to come back and review this. But on that graph, the tidal volume was quiet inspiration and quiet expiration. If I'm going to take in a bigger inhalation, and I'm going to force it, and I'm using my muscles, the SCM, the scalenes, the pec minor, and the serratus anterior, what volume of air am I drawing in? We call that volume the what? Inspiratory reserve volume. So we'll look at that in the graph in a moment. And now I'm going to blow out that breath. What did we say about normal expiration? Are there any muscles involved with normal passive expiration? No, it is a passive process. There's no muscles involved. You push air out because as the diaphragm relaxes and as the external intercostals relax, that contraction, or sorry, that relaxation is what's now going to do what? Make the thoracic cavity's volume go down. And with lower volume comes increased pressure. That increased pressure is not going to push the air up and out and oppose the atmosphere. If you were going to blow a candle out, though, you're going to use some accessory expiratory muscles. And what's going to get involved now? What muscles? Where did we go? There they are, right? You've got your internal intercostals. Even the rectus abdominis can get involved. The external obliques can get involved. The internal obliques can get involved. You're really squeezing down and blowing air out. That amount of air that you blow out beyond your normal blowout is called the expiratory reserve volume, or the ERV. Now, if I'm going to take in my biggest breath ever and blow out as far as I can, let me go to the graph. So we're seeing this, right? It's where we dropped off, isn't it? So I'm looking in the middle of the graph, trying to pull all this together. This amount of volume is what? What do we call this little bit up and down? Tidal volume, right? VT, tidal volume. That's normally about 500 mils. Again, that's that average 25-year-old, 150-pound male that these numbers were averaged upon. But about 500 mils in and out at quiet, ebbing and flowing tidal volume. When you go to take in that biggest breath possible, you are inhaling above your normal. That would be this area here, and that is your IRV. When you go to blow out more forcefully than normal, that is this area. Where'd it go? Over here. That's the ERV. If we take the IRV and the ERV and the tidal volume and we put them together, that gives me from here to here. That's my vital capacity. The capacity of life, vital capacity. It kind of makes sense. Now, even when you blow out as forcibly as you possibly can and you've got your rectus abdominis and all those obliques involved and you're squeezing all that air out, there's still going to be an amount of air left behind. The air left behind will be called the residual volume. You cannot possibly pump all that air out. there will always be air left behind. If I look at the total from the top to the bottom, theoretically, your lungs should be able to hold, on average, again, about 6 liters, 6,000 milliliters. It'll be more for me and less for a shorter person. It'll be more for guys than a woman of the same height. So we'll be looking at that in lab in a week or so. We'll be looking at some charts, and you'll look at your age, you'll look at your height, and you'll look at male or female, and you'll decide what should, or on average, what would your total lung capacity be, and therefore what should your expiratory reserve volume be, and what should your vital capacity be, and then you'll actually measure it. And you'll see how your numbers personally stack up against the average, and it'll be a fun little comparison. Now, what I'm going to share with you is I'm going to post for you tonight. Actually, no, I'm going to wait till Thursday. On Thursday, for those who are here, I'm going to bring in an example of the kind of analysis I want you to do. Okay? I'm not going to post it. It'll be for those who are here Thursday and here only on Thursday. And I will give you a problem set, if you will, and we'll work through it together, and we'll identify these different volumes and talk about what's going on. So that'll be the bonus for those who come Thursday. You'll have that little worksheet that we'll do together. Now, not to memorize these numbers, but just to appreciate them, in lab we'll have you, quote, memorizing these numbers. Now I want you to understand what they mean. What we see is that the vital capacity for males and females, that's pretty much the same, right? 500 milliliters. The difference is in the inspiratory reserve and the expiratory reserve. That would also make a significant difference between the total lung capacity. Again, that 6,000 was that male, right? That's that one person. If it was a female, there would be a significantly less total lung volume. Now, that's not to say some 6'10", right, woman doesn't come in here with more than most guys, but on average. So let's pick up. Any questions about the spirometry? That'd be a spirometry tracing. There are instruments that can do this, and you will be in lab in a week or so doing iWorks. It'll be your last hurrah with iWorks, and I can hear the boos now. Your last chance to work with iWorks. And you'll be blowing into a mouthpiece and actually measuring your inspiratory and expiratory volumes. So there'll be a nice little spirometry study that you'll do in lab. Thoughts, questions, concerns over what we've discussed? Make sure you know the muscles of inspiration, the muscles of expiration, both passively and forcibly, and make sure you know the different volumes that I've discussed related to the spirometry tracing. Make sure you know the basic anatomy of the respiratory system. That should be okay. Make sure you appreciate what's happening down at the alveolus as gas exchange occurs over the respiratory membrane. And we'll be talking more about that now. So pulmonary ventilation. Again, you know that when you're exercising, you need more oxygen. When you're at rest, you need less oxygen. So your body is going to make adjustments to your tidal volume, how much you're breathing in and out at rest versus when you're exercising. You're going to breathe more deeply and more often when you're exercising or when you are in need of more oxygen to your tissues. And so we'll talk about the respiratory rate, the number of breaths you normally take at rest, sitting on the couch watching a TV show, and that is going to be between 12 and 18 breaths per minute. We'll say that's a normal range. Kids are more often. They have smaller lungs, so they need to breathe more often to keep up with their brain's oxygen needs. I usually think of 12 at the low end, and the reason is and this number usually works out for most people, but if you're a healthy young person, 12 is a reasonable place to be. If you're breathing in and out 500 mils every time, right? That's our tidal volume. And you're breathing 12 times a minute, what is your total air that you're moving every minute? Yeah, 6,000 milliliters, right? Which is equal to 6 liters. And that's also the volume of the average male lung, right? So it's kind of a nice number. The amount, in other words, you are breathing in the same volume of air as pretty much your lung can hold. I think of it similarly to how much blood you have in your body, about 5 or so liters. And if you looked at your cardiac output, If you looked at your stroke volume times your heart rate, that is somewhere in the same ballpark of 5 or 6 liters. So the same idea at rest, you're pumping all of your blood around in a minute. At rest, you are utilizing your entire lungs volume. It's just a way of keeping these numbers straight in your head. So you've got the number of breaths, and then you have the amount. This is kind of like heart rate times stroke volume, right? Where now the tidal volume is the amount of air pushed with every inspiration, expiration cycle. It's the same concept. It still requires multiplication. Okay? Some of you don't like multiplication. Okay. So we see a chart like this, easy enough. It's like we zoomed in to the big spirometry tracing, and now we're just looking at that little tiny strip down the middle where we saw that little up and down tidal volume of about 500 mils. Remember, we saw that males and females both have pretty much the same. And this chart will not only tell us how much we're breathing in, but also how often we're breathing, where we can see each cycle of the respiration cycle. So this will give us a respiratory minute volume. We're going to call this V sub E, and the way I remember this is everything, right? E. Why in the world is it E? I don't know. This is the letter you'll see. So VE is the volume of everything that's going in and out of the lungs. And just like we said, it's sort of like cardiac output for your lungs. It is the breaths per minute multiplied by the tidal volume, and the breaths per minute in this equation is F. I don't know why, right? But breaths per minute is F, and the volume is VT, and that gives you your V sub E, which is your total ventilation rate.
This is frequencies.
Frequency. Yeah, that's a good F. Okay. You're right, but I couldn't think of it when I was there. So now what will affect that? What will affect that? Variations, right, in either frequency, thank you, or in the volume will certainly affect your V sub E, right, your overall ventilation. And at rest, there's that 6 liters per minute. So in order to get 6 liters per minute, what's the respiration rate? 12. Even though I know it said 12 to 18 was, quote, normal, in order to get that normal average of 6, we have to backtrack and say, okay, that means that the normal person's breathing in and out 12 times a minute at 500 mils each time, and that's where we get that 6 liters per minute. Okay, so there's our numbers, just multiply together. Remember, this can be mils or liters, just convert by 1,000. Do we have this? It's just like cardiac output, isn't it? It really is the same idea. Now there's another number, the alveolar ventilation. This is how much air gets down to the alveoli. Think with me for a moment. If 500 mils are going into your respiratory system every few seconds, right? 500 mils going in and out. How much of that 500 mils is actually reaching down into the alveoli? Less, right? Less than 500. Because some of that air is stuck in the tube. Some of that air is stuck in the conducting zone. And so there's no gas exchange happening there. So now we need to look more specifically at how much of the ventilation, how much of the air going in and out is actually reaching the alveoli. You've got 300 million of these little air sacs, and we've got to get air down there. Otherwise, it really doesn't matter, does it? Right? The amount of air in your trachea doesn't matter. The amount of air down in your bronchioles really don't matter. What matters is the amount of air that reaches down to your alveoli. So there's always a certain amount of air that you cannot get down to the alveolus. In other words, the air in your trachea, the air down through your primary, secondary, tertiary bronchi that are nowhere near the alveoli, that creates a dead space. No matter what you do, there's going to be air stuck in that dead space. That's going to be V sub D. Okay? So that's your V sub D, and that's going to be about 150 mils. Okay, do some subtraction for me. If 150 mils are stuck in the conducting zone, how much air is actually getting down to your alveoli? 350, right? 500 mils per breath. Only 150 of which is stuck in the conducting area. So only 350 mils are actually getting down to the alveoli. Now, also think about it. The air that's getting down to your alveoli is not all fresh, is it? Right? Is everything getting down to your alveolus fresh air? there's always going to be a mixing of fresh air and expended air that are kind of coming and going through the conducting zone. So there's certainly going to be less oxygen and more CO2. If I measure the oxygen in the alveolar air, there'd be less oxygen than in the atmosphere. And there would be more CO2 in the alveolar air than in the atmosphere. Can we picture why? Does that make sense? Because as you are blowing off CO2, some of that CO2 will get stuck in the conducting zone. And then when you breathe in again, you're bringing in the oxygen of the atmosphere, but you're also adding it to some of that CO2 that was stuck in the conducting zone. So what gets down to the alveoli will be a mix of used air and freshly inspired air. So we can draw this in a cartoon like this. I think it's pretty amazing because when we talk about the structures of the respiratory system, for some reason in my mind, I imagine that the conducting zone contains a larger volume than the alveoli. Right? We think about the trachea and down to the carina where the splits, and then the primary and secondary and tertiary bronchi. There's something in my mind that says there must be more air stuck up in the conducting zone than actually get down to those little tiny alveoli at the bottom of the tree. But in fact, right, 350 out of the 500 mils will get down into, right, the functional regions of the lung. And we can draw it sort of like this. The tube going down would be the conducting zone, the straw, and then the purple would represent a single alveolus as a representation, but multiply that by 300 million. Right, so we have all the conducting zone and then all this huge area of the double tennis court making up for the gas exchange. So that means now if I want to calculate my alveolar ventilation, it's my number of breaths per minute multiplied by the volume of air in the alveoli. That means I'm going to subtract V sub T versus V sub D. What was V sub T? 500. And V sub D was 150. The difference is 350. So how much air is actually going to get down, assuming the frequency is still 12? So if I'm breathing in 12 times a minute, and only 350 mils of each breath is making it down to the alveoli, what is therefore the V sub A? $350 times 12. 7 times 6. Yeah, 4,200, right? 4,200 or 4.2 liters. Okay. Now, when demand for oxygen increases, you're exercising, what's going to happen? We know this, sympathetic, right? You need more exercise, you're breathing, you're going to breathe deeper, you're going to be using those muscles, you're huffing and puffing, you're going to be using those accessory inspiratory and expiratory muscles, and you're going to be breathing more frequently. So what's that going to do to your overall ventilation rate? Whoop, it's going to go a lot higher, isn't it? Because you need to get that oxygen to your muscles. It all makes common sense. We know this from our everyday experiences. We just have to put terms to it. We need to understand the anatomical and physiological terms and apply it to what we already know to be true. So there's our numbers. Now, in this case, the frequency was 20. So that would go from, and here, the tidal volume was only 300. Right? Is everyone going to have a tidal volume of 500? No, every patient's going to be different. Every patient who walks in the room is going to be different. So the average is 500. So the tidal volume may only be 300. That 150 is kind of stuck, right? That's going to be pretty constant within a person. There'll be some variations, but for the most part, that 150 is kind of a good average. You're not going to see that varying very much. So here we see that the whole thing is cut in half, isn't it? 6 liters was the V sub A, or V sub B, everything, and V sub A was half of that. So whatever the numbers are, you would just plug and chug. So we've got Boyle's law. We've talked about Boyle's law already. That was the law that talked about pressure and volume being inversely related. And we know that because volume and pressure are inversely related, that's what's actually drawing air into and pushing air out of the lungs with each and every breath. There are 2 other dead white dudes that I need to describe. Okay, we got Dalton and Henry, also describing different gas laws. Okay, so let me go through each of these, and These are all the, quote, the gas laws, and they're all going to contribute to our understanding of how gas exchange occurs. So I've already told you that at sea level, that our atmospheric pressure is 760 millimeters of mercury. That would also be equal to 760 torr, and would also be equal to 1 atmosphere. Right? 1 atmosphere, 760 torr, 760 millimeters of mercury. Now, each gas in our atmosphere is contributing to the overall pressure. What is the most abundant gas in our environment? Anybody know? Yeah, nitrogen. Oxygen is not the most abundant gas. So about 78%, there'll be a chart coming up, but about 78% of all of our air in the atmosphere is nitrogen. So that means that nitrogen is providing or giving us 78% of the pressure. So we're just going to divide this up very easily. And what's the percentage of oxygen? Anybody know? About 21%, right? So we've got 78% nitrogen, 21% oxygen. That means that oxygen is providing about 21% of that 760 millimeters of mercury. And so Dalton is the guy who came up with this, and it's very easy. You take all the individual pressures of the gases and we add them together, and at sea level they equal 760. Okay, so don't make this harder than it is, it's just addition. So we look at the atmosphere, 760 millimeters of mercury. How's that happening? Well, about 78 or so % of the air is nitrogen. About 21% of the air is oxygen. And a very small percentage, 0.04%, is made up of carbon dioxide. Then you consider if you're in Houston in the summertime, it's much more humid. So there's more water vapor, more humidity, more water is in that. And if you're out in Arizona, then you know that it's much drier air. So the amount of water in the atmosphere is going to vary based upon where you are in the time of year. But the gases are the same, right? 79, 21, and 0.04%. And that's true in Denver, and that's true here. The percentages are not changing as long as you're in our stratosphere down, on the planet. You are breathing the same concentrations. Again, what's changing is the pressure. Now, let's take a look at alveolar air. What did I tell you? The air in your alveolus would have what? Think about it. You're breathing in. As you breathe in air, that oxygen you're breathing in is going to be dropping. As you breathe in, the CO2 is also going down. You're going to have more CO2 down in your alveolus, aren't you? You're going to have less oxygen. You're going to have more CO2. And what about water vapor? What else is the respiratory system doing as you inhale? It's humidifying and warming the air. So you're also going to expect to see more moisture in the air of the alveolus. So let's just take a look. The top row, this is inhaled air in Tucson. This is exhaled air. So let's take a look. Down in the alveolus, what did we say? Less oxygen and more CO2. So we see that to be true. And down in the alveolus, more moisture. Because as we take our air in, we're going to be moisturizing or humidifying that air. As we exhale our air, we're blowing out about 3.7% CO2. A lot more, right? We're blowing out, that's a lot more, isn't it? It's a lot more percentage-wise than what we inhaled. And we're also going to be blowing off less oxygen. We blow off oxygen, right? We take in oxygen. We always have this perception. I mean, what do all the diagrams tell us? That we breathe in oxygen and we blow off CO2. Yes, but is that all we're blowing off? Don't be tricked by that simplicity. As we breathe in oxygen and we blow off CO2, we're also mixing that air, and we're not just blowing off CO2, right? We're blowing a lot of oxygen out. It's just a lot less than what came in. And we're going to inhale CO2 a lot less than what we blow off. Okay, so just look at those numbers. And then if we look at the top, what this is telling me, I'm going to clean this off. What this is telling me is that if there's 760 millimeters of mercury, what Dalton told us and what we need to understand, that 78.6% of the 760 is coming from nitrogen. So 597 millimeters of mercury are attributed to nitrogen. 159 millimeters of mercury are attributed to the oxygen. A very small percentage, 0.3 millimeters of mercury, is contributing to the CO2 and the water vapor. Again, in Tucson, dry air, you're looking at about 3.7 millimeters of mercury. So we add up all the partial pressures and we get 760. Are we okay with Boyle? And we're okay with Dalton. Third guy to look at is Henry. Henry's law says that at any given temperature, the amount of air, amount of gas, is directly proportional to the partial pressure of that gas. So at any given temperature, the amount of a particular gas, and I didn't say this, in a solution, is directly proportional to the partial pressure of that gas. Let's put Henry into a picture, figure this out. So here, we're in a pressure cooker. And under pressure, gas molecules, we're pushing down, we got a lid on there, it's clamped down. And during pressure, gas molecules are forced into the solution. So we're forcing the gas molecules into the solution. And when we're not under pressure, then the molecules can move out, if you will, away from that. So this is what's going on in a soda can. Right? When you open up a can, there's CO2 that's released. The carbon dioxide comes out of solution. Right? If you look at a clear bottle, you don't see the gas bubbles, right, in a closed soda bottle. But then when you open it, all of a sudden you see that CO2 released out of the solution. It was magically appears out of nowhere, right? And so what happened is that under pressure, that CO2 was pushed into the solution. It's carbonated water. And as you release it, it comes out. Now, you leave your soda open for a few hours and it becomes very flat, right? The CO2 has escaped. There's no more pressure keeping it in. If you don't keep your soda tightly capped, it will get very flat. And here, when we make the soda, we're putting that CO2 in under pressure. It reaches an equilibrium, right, inside, underneath that cap. Pop the cap, everything comes out. After a while, that CO2 is going to be released because there's nothing holding it in place. So this is Henry telling us about the percentage of gases that are being pressurized into a fluid. So we've got Boyle, Dalton, and Henry to keep track of. Now here are some numbers. This is back to millimeters of mercury. I want to go back for a moment and look at that table. What is this telling us? In the atmosphere, there's 159, 160 millimeters of mercury contributing to the oxygen. Okay, so that's the partial pressure of oxygen in the atmosphere. Keep that number in mind as we look at this. External respiration, right, bringing air down. We're already down now to the lungs. We're going into the blood. So in arriving deoxygenated blood, venous blood, right? Let's get a picture of this. This is venous blood. It's coming back to the lungs. We've already dropped off oxygen. We're coming back into the lungs. It's deoxygenated blood. It's blue blood. And in the capillaries of the alveolus, this blood is going to get reoxygenated, isn't it? The alveolar PO2, pressure of oxygen, P sub O2, the pressure of oxygen, is 100 millimeters of mercury. And because there's a concentration difference, because of pressure difference, which way is oxygen going to go? There's 100 millimeters of mercury of oxygen in the air, in the alveolus, and there's only 40 millimeters of mercury of oxygen in the alveolar blood, or in the capillary blood, the venous blood. So which way is oxygen going to go? From high concentration to low concentration, or from higher pressure to lower pressure, because what did Henry just tell us? Pressure is going to help push gases. Think of it that way. So we're going to see now at this point, oxygen is going to diffuse into the blood. What did it do to get from the blood to the alveolus? What did it have to cross? The respiratory membrane, right? Are we with me? So to get from the alveolus into the blood, I had to cross over that triple membrane, if you will, that triple layer. Now, what about CO2? CO2 coming in the blue blood, in the deoxygenated blood, coming back to the lungs, has a pressure of 45. In the alveolus, it's 40. So which way is CO2 going to move? These gases are going to move independent of each other, and they're going to move based upon the pressure differences and the concentration differences. So what do we see now? CO2 is going to go from the blood into the alveolus. It's going to be moving in the opposite direction of oxygen. It too has to move across that respiratory membrane. And as a result, CO2 will diffuse into the alveolus from the blood. So let's look at a picture of this. Pictures are a lot easier. So now you're looking at a capillary next to an alveolus. You've got this going on millions and millions of times within your lungs, happening every few seconds. Pretty amazing process. And here are the numbers for you to capture. Blood is coming in from on top. So we've got the blood coming in, is coming in a pulmonary what? Coming into the capillaries of the lung. It's coming in from the right ventricle of the heart. Is traveling there through a series of pulmonary arteries. Thank you. Okay. Remember, these are the blue arteries. And that blue blood's coming in. It's going away from the heart. Coming in. And we get down to the little pulmonary capillaries. And every little capillary is right next to its own little alveolus. Cute little arrangement. And what we see now is that the O2 coming in has 40. And the CO2 has 45. Those are numbers you need to appreciate. By appreciate, I mean know. And then in the alveolus, we've got 140. So what's going to happen? Gas is going to move, isn't it? O2 is going to come in, CO2 is going to go out. So now what does the blood have as it leaves the lung? It's going to bring the pressure of oxygen up to 100, and it's going to lower the pressure of CO2 back down to 40. Do you see what's happened with the numbers? Right? We lost CO2 to the alveolus. We gained oxygen from the alveolus. And as a result, the red blood going back to the left atrium of the heart is going to be a PO2 of 100 and a PCO2 of 40. Let's put this all together. Now, that oxygen that blood is going to go back to the left atrium, go down to the left ventricle, and get forcibly pumped up and out of the aorta. And that blood is now going to travel to all of the tissues in your body. And now we're going to start talking about internal respiration. And that blood is going to reach your tissues, right? Internal respiration, by the time it gets down to the tissues, it's going to have dropped a little bit. Remember it was 100 when it left the lung? It's going to drop a little bit. It's going to drop down to 95. Not a big deal, but it's going to drop down. We're going to lose a little bit of oxygen along the way. The interstitial fluid has oxygen at around 40. Are you picturing this? Where am I? What environment am I in when I'm talking about 95 oxygen and 40 for the tissues? Are you with me? Are you picturing where I am? I'm down in a capillary bed in your muscles. I'm in a capillary bed in your brain, right? I'm in a capillary bed where you're dropping off oxygen. And when you drop off oxygen, can you picture it now? Think about the lymphatic system. Think about the cardiovascular system. You got these capillaries. They're coming in red. Across the capillary membrane, we're going to be dropping off oxygen into what? Into the tissues. But before I can go from the capillary into the tissue, where must that oxygen go? Into the interstitial fluid. Do you agree? Remember, that's fluid and gas exchange and nutrient exchange and hormone exchange and all that exchange stuff that's happening at the capillary. Oxygen's going to leave the blood, go into the interstitial fluid. Now, oxygen is sitting there in the blood at 95. The interstitial tissues have a PO2 of 40. So which way is oxygen going to go? It's going to jump out, right? It's going to move out by diffusion. This is going to be normal old, passive, no energy involved diffusion. Where does that oxygen keep going? Why is it always 40? Why does oxygen keep being pulled out of the capillaries? The cells are using it, right? Because that interstitial fluid has oxygen, and all that oxygen is going to be now taken up by the neighboring cells. So you've got to picture this neighborhood of cells that are sitting right in proximity to this interstitial fluid, and all those cells are demanding oxygen. So the metabolism of each cell is pulling that oxygen into the cells. So that's going to diffuse the oxygen from the blood into the tissues at the capillary spaces. What's happening with CO2? Whoop, flip it. What are all the cells doing? They're busy doing their metabolism. Every cell's making CO2. That CO2 is leaving every cell and sitting at the bus stop, right? Kind of the interstitial fluid is like the bus stop. And what's going to come by? Blood's going to come by the bus stop, and that blood arriving in has a PCO2 of around 40. The tissues are going to have added more CO2, so the interstitial fluid is going to be around 45. So CO2 is going to go which way? It's going to jump on the trolley, right? It's going to jump onto the blood and diffuse from the tissues back into the blood. This is either making total sense or is completely garbage, depending upon your mindset right now. Can I ask, if this is making sense, put thumbs up? Can you picture it? I need you to be able to picture it. You need to be able to picture this. If it's just facts and numbers, you're not doing well right now. I need you to be able to picture this, and we'll look at pictures to try to pull all this together. Let's use this one. I'll assume the people who didn't have thumbs up are in the second category, right? Not quite sure that you're getting this. Let's try to put a picture on it. Blood's coming into your capillaries. This is very simplified. Blood's coming into your capillaries with a pressure of 95 millimeters of mercury. CO2 is coming in at 40. Where did those numbers come from? A while back we said that oxygen was 100, right? And it dropped a little bit down to 95. Comes in, and every capillary is going to go by a series of cells. So here we've got some muscle cells, some epithelial cells, just all sorts of kinds of cells, depending upon what tissue we're in. Oxygen's going to go out into the tissues. Why? Because this interstitial fluid, on both sides, on all dimensions, not just on one side, but there's going to be leakage coming out of these capillaries. O2 out, moving toward a lower concentration. CO2 coming in. That oxygen and CO2 exchange is going to make that arterial blood now be venous blood. That venous blood returning back, the numbers have changed. Now the PO2 is 40 and the PCO2 is 45. If you go back, those are the same numbers I said that were arriving in the lung. Right, so this blood's going to go back to the lung and pick up more oxygen, and we're going to go around and around. These numbers, there aren't that many of them, but there's a few that you need to appreciate. What do we think? Okay. Fight through it. Look at pictures. Listen to this again if it's not making sense. Pictures are really good in your martini book. So let's put a few more numbers to this. Every 100 mils of blood leaving the alveoli is going to carry about 20 mils of oxygen. What that means is that there's 20 milliliters of oxygen. It's not very much is what I want to say. Only 1.5% of your oxygen is dissolved in the plasma. Most of your oxygen is hitching a ride on what molecule? Hemoglobin. So the vast 98% of all of your oxygen has hitched a ride on that hemoglobin molecule. 1.5% or so is dissolved. Oxygen does not dissolve in water very well. Okay? So most of it is attached to the heme group. Remember, the heme group is the little iron-containing group in the center of each hemoglobin molecule. And how many oxygens can each hemoglobin carry? 4. Okay, good. If the hemoglobin is carrying all 4 oxygens, then we'll call it oxyhemoglobin. Okay? So oxyhemoglobin is hemoglobin carrying oxygen. It's not hard. Just put it together. CO2 can bind a little bit, But the real culprit here is carbon monoxide. We hear every year about people getting carbon monoxide poisoning accidentally. They're getting carbon monoxide. Carbon monoxide is a odorless, invisible gas that can bind to the heme groups, and it binds stronger than oxygen. So if one is in a higher concentration of carbon monoxide, then they're no longer able to transport oxygen. So if a person is in an environment with a lot of CO, it will bind up all their heme groups, and eventually these folks will die of basically asphyxiation. They no longer have the oxygen going to their brain, so they'll faint, they'll go unconscious, and they'll die as a result of no oxygen. Get them into fresh air, and they can recover if you do it fast enough. Here's a cartoon of a hemoglobin molecule. As a reminder, there's 4 parts to it. Each part can bind 1 oxygen. And the little Saturn in the center is the heme group, and the very red dot in the center is an iron molecule. Okay, iron 2+. So we need to look at a curve here. We need to take a look at an oxygen hemoglobin saturation curve. If hemoglobin is 100% saturated, how many oxygens are bound to hemoglobin? How many can it hold? 4. So if a hemoglobin molecule is 100% fully saturated and it has carried as many oxygens as it can, it would have 4. What if hemoglobin is only 50% saturated? Then it's only carrying 2. If it's carrying 25% saturation, 1. Okay, so we got that idea, right? 100% saturation is carrying 4 oxygens. 50% saturation would be only carrying 2. And we're going to look at a curve that shows us this relationship between bound oxygen and a couple different conditions that can change this binding. We're going to see an S curve because what's going to happen is that as one oxygen binds to hemoglobin, it actually makes it more likely that a second one will bind on. And then when a second one is bound on, it'll be more likely that a third one will bind on. So there's this affinity that increases. Hemoglobin is more than 90% saturated with a PO2 above 60. What was our number? What did we say? When air comes into the alveolus, we had a number of 100, right? PO2 was 100. And we said that as that PO2 travels through the tissues, by the time it gets to the tissues, it's dropped down to 95. What am I telling you here? Anytime that the numbers are greater than 60 millimeters of mercury, hemoglobin will be 90% or better saturated. When you go to the clinic and they put that little meter on your finger, right? And they tell you your SATs. What are they looking for? They're looking for the percent of your hemoglobin that is saturated with oxygen. And if your SATs, if your O2 SATs are dropping low, something's going on, right? But most of us would be really high, right, in normal. So that's the connection clinically that you'll see as we go through this. So that means that at normal millimeters of mercury of oxygen, as it comes into your tissues, at 95 millimeters of mercury, your hemoglobin will be about 97% saturated. If it's only 40, it'll be 75% saturated. What does that tell me about the oxygen as it goes through the capillary of the lung? If it's going in at 95, what's it coming out at? Go back and look a couple pages back. Right? The oxygen's going in at 95 and leaving at 40 millimeters of oxygen, yes? So look what those numbers mean. As long as at 95, we're 97% saturated. At 40, we're only 75% saturated. So that means that basically when hemoglobin traveled through the capillary, it was carrying essentially all 4 oxygens, wasn't it? Right? It was 97%, essentially 100% saturated. When it left, it's at 40 millimeters of mercury of oxygen, and I'm telling you it's only 75% saturated. So how many of the 4 oxygens just jumped out into the tissues? 1. 4, 100% essentially. When I'm leaving the alveolus, I'm leaving with only 75% saturated, meaning I've still got 3 oxygens attached to my hemoglobin. I'm only dropping off 1 out of 4. Tell me what situation makes sense to you, or how does that help you understand how one can hold their breath? Hold your breath, right? Blood's going around your body. Every time it goes through, it's dropping off 1 oxygen. How long can you hold your breath? I mean, people can do this for a lot longer with a lot of practice, but the average person can hold their breath for, if you really push it, a couple minutes. Right? So what does that tell us? The reason we can hold our breath for a couple minutes is that there's still oxygen in our blood. It's not all being dropped off in the capillaries. It's all not being, right? We're not replacing or losing all of our oxygen each and every time we go around. At PO2 of 20, this is what would happen in an active muscle tissue. So as you're going through the muscle, you're going to be dropping off more oxygen. You're going to drop down to about 20% saturated. So you couldn't hold your breath as long when you were exercising, could you? Because more oxygen is being pulled off your hemoglobin as you're exercising. There's a greater oxygen demand. So as your blood goes through your muscles when you're exercising, you're going to be dropping off more oxygen. But at rest, just sitting there right now, you're only dropping off about 1 oxygen every time the blood goes through the capillaries. So here's that curve. It's kind of like an S-shaped curve. Okay, I'm going to kind of over-exaggerate, kind of an S-shaped curve. And what this is saying is that normally 95 millimeters of mercury, way up there. That's where your O2 usually enters into your tissues. And at that point, your blood is about 97% saturated. And then as it leaves, we say that it's about 75%. And that is equal to 40 millimeters of mercury, or millimeters of mercury. So you're going in at about 97%, 100 or so, 97, and you're coming out at 75% saturated at about 40 millimeters of oxygen. If we were in an active muscle, we'd be way down here, about 20% saturated. We're down to about 15 millimeters of mercury. We're getting pretty low. So this little S curve will help you appreciate the saturation curve. We're talking about hemoglobin saturation versus the millimeters of mercury traveling through your blood. Now, what can affect how much oxygen can bind to your hemoglobin? There are 2 factors that I want you to know about, and one other molecule that I'll talk about, and this was attributed to Bohr. So this is our 4th guy to keep track of. We've got Boyle, Dalton. We talked about Henry and gases being pressure of gases, and now we've got Bohr. And Bohr said that as the pH decreases in the blood, that the saturation curve will shift to the right. I'll show this to you in a moment. And that as the pH increases, the saturation curve will shift to the left. So more acidic to the right. More acidic to the right, right? pH decreases is more acidic to the right, less acidic to the left. Also, in addition to pH, temperature can have an effect. Higher temperatures will lead to hemoglobin releasing more readily. That is, it's also shifting to the right. So higher temperature and lower pH shifting to the right. There's another molecule that is naturally made by your own red blood cells called BPG. Long name, 2,3-biphosphoglycerate. This is a molecule that is generated from glycolysis in red blood cells, and what happens is that more of this PBG will lead to more hemoglobin releasing oxygen. You produce less of this with older red blood cells. So let me show you this curve. What is your normal pH of blood? 7.4. So that means that the normal S curve would be at 7.4, normal pH. But what happens if our pH of our blood drops down to 7.2? More acid, more acidic, shifts this curve to the right. Okay, so when I said to the right, imagine this curve being shifted to the right. All the numbers are shifting. And if instead the pH went up, less acid, to pH 7.6, then you would see everything shifting to the left. So we'll see how pH, what's going on here in a few minutes, but look back and think about this curve. Increased acid, increased temperature, shift to the right. Same thing's going to happen. This was pH, and this is what happens with temperature. You still see the S curve. Your normal body temperature is around 38 degrees Celsius. So we see the normal S curve in the orange curve. At higher temperatures, 43 degrees, you got a good fever. Again, shifting to the right. Higher temp, lower pH, shift to the right. Lower temp, higher pH, shift to the left. But in normal physiology, right? I mean, you're not going to have a body temperature of 20 degrees very often, right? It's pretty dramatic change. So most of the time, we're sitting there right at our body temperature of about 38. Part of it, trying to get rid of and increase their heart rate. Yeah. Hold on to that. We'll come back to that as we get through this. So we just talked about oxygen. Let's just talk about CO2 for just a moment, because what do we know about CO2? This is important as we think about this. I'm going to jump ahead a little bit so that as I go through this, you're appreciating this. CO2 and water combine to make carbonic acid. So in your blood, the more CO2 there is, the more acidic your blood is. And as a person is having respiratory problems and they're not able to blow off as much CO2, their pH in their body is going to go down, right? So as a person is not able to blow off CO2, that CO2 is going to accumulate because all of your tissues are still metabolizing. You're still making waste product CO2. And as your CO2 levels rise, your pH is going to go down because you're making more acid. So let's think about this. So as your tissues, all of your cells in your body are doing their internal respiration. Internal respiration means they're taking in oxygen and producing CO2 as a waste. That CO2 is accumulating and is traveling back through your venous blood. That CO2, though, travels in three different ways. Number one, it does dissolve a lot more than O2. CO2 is able to dissolve in water or in plasma much better than oxygen. Number two, CO2 travels also on hemoglobin, not the same place as oxygen. Okay, I want you to understand this. Hemoglobins can carry how many oxygens? 4. What molecule inhibited or got in the way of oxygen binding? CO, carbon monoxide. CO2 also travels on hemoglobin, but at a different place that does not compete with or interfere with oxygen carrying. So about 23% of your CO2 is bound to parts of the hemoglobin molecule. And then finally, there is some of the CO2 that is converted to this carbonic acid that I'm referring to. In fact, that's where most of it is. Most of the CO2 is actually forming acid in your body. So we're going to see that CO2 has a dramatic effect on the pH of your blood. Too much CO2 drops your pH, and now you are said to be acidotic. You're making a lot of acid, you're acidotic, or maybe you're said to be acidemia. You have a lot of acid in your blood because, again, CO2 makes carbonic acid, drops the pH of your blood. The enzyme that does this, the conversion of CO2 to carbonic acid, and here's the chemical for carbonic acid, H2CO3. So when you see that, that's carbonic acid. And the reaction that does this is an enzyme called carbonic anhydrase. It has the same, carbonic acid is made by carbonic anhydrase, and you'll see that enzyme abbreviated Ca. So if you see Ca anywhere in a diagram, it's referring to this enzyme, carbonic anhydrase, and here's the chemical equation for that. CO2 plus H2O makes carbonic acid. Now carbonic acid then breaks down, and look what it does. It then dissociates or breaks down into HCO3, that's bicarb, and H+, hydrogen ion. What we're going to see in a moment as well is that, in a picture, is that that bicarbonate is then shifted out of the cell with a chlorine ion, and that's going to be called the chloride shift, and the hydrogen will grab onto the hemoglobin. And what happens now is that the hemoglobin actually will act as a buffer. So you've got all this acid being produced. And all that acid could cause tremendous issues with your blood pH if there wasn't a lot of buffering going on. So you have a lot of buffers also in your blood. So let's take a look at a picture of this. And honestly, I think this is the most difficult concept in this conversation, is right here, trying to understand this bicarb chloride thing that's going on. So you may have to stare at this a little bit longer. So what's happening? CO2 is coming from your tissues, and it's going to be released and grab a ride in your bloodstream as the blood goes through the capillaries. 7% of that CO2 is going to be, not 7, 7% of that CO2 is going to be in solution. It's going to dissolve in the plasma. 23% or so of that CO2 is going to travel around attached to hemoglobin. When CO2 is attached to hemoglobin, we call it carboaminohemoglobin. HbCO2, because it has CO2 on it. Before we had oxyhemoglobin, now we have carboxyhemoglobin. And then the majority of that CO2 will be riding through the blood, converted by the enzyme carbonic anhydrase to carbonic acid. Now what's it doing? Then that carbonic acid spontaneously dissociates to H+ and bicarb HCO3-. The H+ is now going to catch a ride on the hemoglobin. The bicarb is going to leave the cell, leave the red blood cell, and in exchange for a chloride, Cl-. You have an HCO3-, a negative ion leaving, and a negative ion coming in. So the charge isn't changing. Bicarb out, chloride in. And this is called the chloride shift. You'll see what that's about as we finish this up. Questions? Not about chloride shift, because give me a second to finish that story, but anything else? Any hanging ideas here? Who are my respiratory therapy students? How am I doing? Am I doing okay? Making good sense to you. Now turn around and teach everyone else, right? Because you've been hearing this, right? You've been hearing this in different ways, and so the story makes sense to you in more complete ways than everybody else in the room. Okay? Yes?
I just have a question.
They stick together. Isn't this weird?
We do everything. Okay, when you were talking about 2,3-BPG,
Yes.
we were taught that it's DPG. Is that
Bi versus di.
Okay, same.
That's what just the two.
Same thing, right? 2, 3. So some books will say diphospho and bisphospho. Bi, di, 2, same thing. Okay, good catch though. Same thing. Anything else? Okay. So respiratory gas equilibrium. PO2 and PCO2, the pressure of oxygen and the pressure of CO2, are stable in alveoli, and tissues. There's a balancing act, right? There's an equilibrium. There's a balancing act that goes on here. And when that equilibrium is disrupted, or that equilibrium is disrupted, when tissue oxygen levels when tissues start demanding more oxygen. Kick this out, start it back up. So when your tissues start screaming for more oxygen, then this equilibrium is changed. So in order for you to make up for that oxygen demand, right, your respiratory rate's going to go up, you're going to start breathing faster, and you're going to increase the depth of your breathing, right? You're exercising. So you're going to be breathing heavier and more often, deeper. That's going to increase your overall what? If you increase your tidal volume and you increase the frequency, what is being increased? What was the equation? V sub what was equal to the frequency times vital V sub E, right? The volume of everything, right? That's the way I think about it. So the V sub E is going to be increased. So your respiratory rate and your tidal volume are going to go up. V sub E is going to go up. Now, without that increase, the alveolar wouldn't go up, blood oxygen levels wouldn't go up, tissue levels would not go up. We've got to keep these up, right? If you're exercising, you've got to increase your oxygenation to your tissues. So we're going to increase our V sub E, we're going to increase our V sub A, we're going to increase our millimeters of mercury of oxygen going to the tissues. If you weren't to do that, then what would happen? If you did not increase your ventilation rate and your tissues were grabbing more oxygen from your blood, you would quickly become hypoxic, right? You would not have enough oxygen, or hypoxemic, not enough oxygen in your blood. And wherever there's more oxygen, there's always less CO2. Wherever there's always less O2, there's more CO2. So if you're hypoxic, this rule will get you through 99% of the time. If you have less oxygen, that means your body has more CO2. If your body is accumulating CO2, then what's happening to the pH of your blood? It's going down, right? You are becoming acidotic. So keep that in mind. That'll get you through everything I'll be dealing with. That'll get you through all the situations. So stare at this figure for a moment. Take a look at this. And what's happening is it's showing us O2 pickup from top left to top right, the pickup and the delivery of oxygen. And then the bottom is showing you how it is that CO2 is picked up and then delivered to the alveoli. So on the far right-hand side, this is your tissue. And on the far left-hand side, this is your alveolus. Okay. And we're looking at the incoming oxygen, transport to the tissues, kicking out of CO2, and then the transport back. What we see ultimately, O2 in and CO2 out. Right? That's what we always see. Again, don't make it that simple. It's okay to start there, but don't think that that's exclusively what you're taking in or exclusively what you are blowing off. Let's watch a little video. This will be fun.
To get energy. And breathe out carbon dioxide, a waste product. When you inhale, your diaphragm and rib muscles contract, increasing the volume of your lungs. When you exhale, these muscles relax, decreasing the volume of the lungs. When the lungs expand, air pressure in the lungs drops, causing air to flow into the lungs. When lung volume decreases, air pressure increases, causing air to flow out of the lungs.
According to who? Boyle. Right? Boyle's law.
Air enters the nose or mouth, moves down the trachea, and goes into the two bronchi. Air moves down smaller and smaller bronchioles until it reaches a tiny sac, an alveolus. Each alveolus is surrounded by capillaries. Oxygen diffuses from the alveolus to the blood, and carbon dioxide diffuses from the blood to the alveolus. As blood flows through the capillary, it becomes rich in oxygen. In the blood, oxygen diffuses into a red blood cell and binds to hemoglobin, a protein made up of 4 subunits. One oxygen molecule can bind to each subunit. Oxygen-rich blood flows from the lungs to the heart. Which pumps this blood to capillaries all over the body. Here, we see oxygen diffusing from a capillary's red blood cells into a muscle cell. Oxygen is used by the cell's mitochondria to produce ATP during cellular respiration. Carbon dioxide is released. How does carbon dioxide leave the body? Carbon dioxide diffuses from cells into capillaries. Some carbon dioxide stays in the plasma, the liquid part of the blood.
7%.
Most carbon dioxide, however, enters red blood cells. Some carbon dioxide binds to hemoglobin.
23%.
The rest is converted to bicarbonate, which diffuses into the plasma. This oxygen-poor blood flows back to the heart, which pumps it to the lungs. There, carbon dioxide diffuses from the plasma into the alveolus. Bicarbonate enters red blood cells and is converted back to carbon dioxide. Carbon dioxide is also released from hemoglobin. Carbon dioxide diffuses out of the red blood cells into the plasma and into the alveolus. When you exhale, air flows out of your lungs. And that's how you release carbon dioxide, get oxygen, and keep on running.
Okay, it's always good to hear somebody else say it in a different way. Okay. Last couple of things, some clinical connections here. We'll also be talking about these in lab. There are a number of pulmonary diseases that I want you to appreciate and how they affect the lung and how they affect the function of the lung. Compliance refers to how stretchy, if your kids are compliant, right, they do all the right things at all the right time, right? So a compliant child or a non-compliant child. So the compliance of the lung refers to that as you are expanding your thoracic cavity, the lungs go with it and air is able to come in. So a highly compliant lung is one that will expand as needed and will fill up with blood as needed. It has the correct elasticity and it has the correct resilience to counter that. It also would be influenced by the flexibility of your chest wall. Obviously, if you can't inhale, then you won't be able to expand the lungs to their full capacity. And remember we talked about how there's that intrapulmonary pressure. That's the pressure, the differential that allows air to come in. So let's take a look at compliance. Again, a balloon would be rather compliant. Have you ever had one of those real stubborn balloons, those really small balloons, right? And you're trying to blow air in and it kind of resists back. So that would be not as compliant. So a balloon that's easy to expand, when you blow into it, would be a very compliant balloon. One that is more resistant to the airflow would be a balloon of less compliance. So normally, the muscle activity in order to do your normal external pulmonary ventilation is going to account for a very small percentage of your overall energy. So at rest, only about 3 to 5% of the energy that you're using up right now is involved with breathing. But what if you had to breathe harder, right? If you, because of lack of compliance, you're trying to blow up that very tight balloon, and you've got to work your muscles harder to do normal breathing, then now the amount of energy necessary to maintain normal respiration would go up well above 3 to 5%. It's going to be much more difficult. You're going to spend a lot more energy breathing than you are other things, and that's going to now make a deficit to your normal other functions of life. So we think about resistance. Increasing resistance is also a problem. If you have bronchoconstriction, if you're narrowing down your bronchioles, like what happens in asthma, when you've got some sort of obstructive disorder, and it's like you're trying to fill up something through a straw, that is more narrow. There's a constriction, an obstruction of the airflow, and that's going to increase the resistance as well. So let's talk about a couple of conditions that you've all heard of. One, there's a whole group of disorders collectively called COPD, chronic obstructive pulmonary disorders or diseases. And under this, one of them, the most common one of these would be asthma. Anybody here have asthma? Anybody deal with asthma? Know somebody with asthma? It's not that uncommon. So asthma, what happens is that there is a problem with an irritation and a swelling of the airways that occurs by different triggers, and that can lead to a tremendous obstruction to the air. This is going to make it more, it's like you're breathing in and out of a straw. Have you ever tried that for a while? Right? Just your normal breathing and do it in and out of a straw. For a couple, for a few seconds it's okay. But after a while, you really want to get more air out and you really want to get more air in, and you get really frustrated. So imagine now a person with asthma feeling that same thing. So it could be an increase in mucus, it could be an increase in inflammation, it could be brought on by toxins, allergens, all sorts of things, or even exercise can bring on asthma. Bronchitis is another form of a COPD condition where now you've got long-term inflammation of the bronchial linings. This produces too much mucus. These folks are hacking all the time, right? They get all that mucus, and that is going to clog the airways. Again, imagine something is obstructing the airflow in and out through the conducting zone. Smoking, a great reason for someone to have chronic bronchitis, but not completely. Other people will have other infections of the lung that can lead to chronic bronchitis. Now, what happens, that chronic bronchitis, subsequently, there can be some heart failure that can go to this, and we'll talk about that as we get into the lab a little bit. But swelling and bluish skin, if you're not getting enough air in and out, that can lead to someone being more blue, right? Because you have low oxygen levels. You've got low oxygen levels, but you also have high CO2. Right? The third is emphysema. So emphysema is also sitting in, now emphysema is kind of a funky disease, but we're going to throw it here right now for COPD. And in this condition, this is a chronic progressive condition, and what happens is the alveoli slowly begin to break down. So rather than having 300 million little tiny air sacs, you have fewer. Now at first that doesn't seem like a problem. What's wrong with having fewer but bigger alveoli? And somewhere in middle school, you learned about the surface area of a sphere. And you learned that the collective surface area of a bunch of small spheres is bigger than the collective surface area of one larger sphere. So there's more surface area to little alveoli. Big alveoli, although they're bigger, have a less surface area. There's less gas exchange going on. So a person with emphysema has a lot less of that tennis court for gas exchange to occur. So those are going to be obstructive disorders. And then I want to get into, actually I'll do this next time. So what I'll do is next time, there's not a whole lot more. I'll finish up with a few more ideas on the respiratory system before our little review session on Thursday. So I will have a little bit more on respiratory on Thursday. I will record that portion, but I may or may not record the rest of the review session that I'll cover with you. If you have any questions or concerns, come on up and see me. If it's in the exam room, it wouldn't be. We have multiple of those. So I got to figure out why in the world.